From the floor, John Bridgewater.
Thanks very much. Question for Arezu. Lovely stuff. Very, very nice indeed. And excellent that you'll be able to get some prognostic data out of it. Actually, for me, prognosis means nothing. The patient will get the treatment anyway. What really is meaningful for me is predictive. I need information to tell me what to give the patient when. How far have you got with that?
Yeah, actually, that's true. Thank you for the question. AI these days is using in many areas in clinical field for prognosis, diagnosis, even for treatment response prediction, and it depends where we need to use AI. And something which is interesting even for us is that we not only want to predict the risk of recurrence in patients, but when we try to explain AI answers, we can see how AI is looking at to the tissue, how it's extracting information about recurrence, and this usually helps us to hypothesize new things about the cellular composition or about the tumor microecosystem, and usually this is something that helps us to go further, and maybe we can validate our hypothesis later in a protein level, maybe in mice model, and so far. And usually what we like and what we are doing is not just addressing some tasks like diagnosis, prognosis, or predictions, classification, stratification. We want to know, and usually I see that many clinicians are interested to know what is behind AI and how AI is looking at the tissue. If the way that AI is looking at the tissue is different from human eye, and this is something that we try to understand. We want to explain AI. We want to see how AI is interpreting its results, and it helps us. This interpretation usually helps us to hypothesize new ideas and new challenges comes, of course, and this is something that we are working. I hope that we can. Thank you.
I think Professor Kendall's scared about his job, so he can have a question.
Absolutely. Sorry, a couple of quick, I guess they're methodological questions. What feature extractor is your current favorite? I mean, is it a flavor of foundation model?
Yeah, exactly. We use foundation models, different foundation models.
Which one's your favorite at the moment? Because I know there's tons of them.
Yeah,
I don't know which ones to
actually,
choose.
what we've tried so far for prognosis, which was 14 different foundation models we have tested so far from Mahmoud Lab and from the US, and the most interesting one for us was Optimus. If you know Akin company from France, they are built this AI model, Optimus. It has different versions, and it's working very well for a prognosis, hopefully, and this is something that we are using right now.
Yeah, and is the backbone, the framework, is it a variety of multiple instance learning that you're using?
Yes, exactly, it's multiple instance learning.
Any particular flavor of that?
Yeah, actually, what we see is that we have a different foundation model, of course, and some of them are looking at a slide, completely whole slide at once, and some of them are extracting patches and looking patch by patch, and sometimes in some tasks, looking at whole tissue is more useful for us, but sometimes looking at and zooming in different patches can have better results for our work. It completely depends to the task and also to the based on the heterogeneity of cholangiocarcinoma and HCC, maybe we think that looking at patches can have better results. I mean, focusing on regions and analyze region by region sometimes is more useful based on the heterogeneity of the cancer type that we face.
Okay.
Thank you.
Last one then. Are you having to do any sort of pre-processing correction of staining, or is the foundation model well trained enough to compensate now?
Yeah, of course, one of the biggest challenges is that we have different scanning environments, different staining types, as you know, from different centers. Usually for France, it's Zafran we use for staining, which is completely different. But hopefully, computationally these days, we can pre-process and we can solve this problem. And even in our project, which is published in 2026, we had very, very good results when we tested and evaluated our AI model on an independent external cohort, because one of the biggest challenges in AI models is that we train an AI model and AI is working very well on our data, but when from another center we receive slides, we see that the model is not working and it fails usually. But hopefully we can handle it computationally, it's possible, and it's working very well with new foundation models.
Super, thank you.
Thank you.
Brian.
Brian Davidson, I'm going to ask you the two questions I thought Tim was going to ask. The first thing is that you mentioned nothing about the intro into your pathway. So what samples you put into the pathway is going to predict your outcome, and you said nothing at all about how you're going to sample. And as you know, one of the bits about an expert pathologist is that they take the specimen and they spend a long time deciding where they're going to sample the information, which is effectively the indoor to your model. So first of all, why did you not mention anything about that?
Yeah, of course, yeah, it's really important what we are doing and what my colleagues actually are doing in Harimondo Hospital to collect HNE slides firstly, and more importantly about Xenium spatial transcriptomic, which is by far a very, very difficult experiment to do, very, very expensive experiment as well to do.
But just come back to the sampling.
Yeah.
So do you need an expert pathologist
Of course.
to identify the samples to put into the model?
Putting the model, yes, this is something that we...
So it's a model that could only be done in an expert pathology centre, because in some ways you're developing a model and it's a question of whether or not it's generalizable. And the answer to that is it's certainly not going to be generalizable unless if you're going to need somebody like Tim to put the information into the model.
Mm-hmm. Yeah, that's true. Actually, we look at this, when we want to select samples, we have 2 points of view. Firstly, computationally, we try to check the quality of samples. So in this case, we are excluding some cases. And the other hand, yes, our pathologists, Professor Kallegaro, usually they select samples, but they try, when they are selecting samples, cover various cases with the highest heterogeneity, because some cases are immune enriched, for example, some others no. So we try to cover many, many cases as much as possible, different conditions,
I
different
understand
staining.
that. My second question for you is the diagnosis of the metastasis. So you're talking about usually here cancers of unknown primary separated from cholangiocarcinoma. So how do you diagnose a cancer of unknown primary?
This was actually a work in our team. It has done, and of course, I've not been completely involved in that. But I know that they built an AI model which trained on a huge data cases from different centers, and now the model can extract features to identify which.
No, I'm not talking about the model. I'm talking about the gold standard, because the model is trying to reproduce a gold standard. So, what is your gold standard for the diagnosis of a cancer of unknown primary?
Actually, I'm not involved completely in this case, but I know that AI is kind of using for diagnosis, but maybe I'm not completely aware about how it's using these days. Thank you.
Could I just ask how many different layers of information the AI can integrate? Do you imagine, or would you go forward after this and try to integrate spatial metabolomics, for example? Mm-hmm. How many levels of information can be?
Yeah, okay. Actually, yeah, it's a good question, thank you. And it completely depends to the AI model, because firstly, it depends to the data. It's sometimes image, it's sometimes gene expression, RNA data, or any information, clinical variables that we integrate to the AI model. And based on what we have and based on what we want, finally, we need to engineer and choose correct AI model. And the answer is yes, we can involve many factors here, but we need to engineer our AI model to be compatible with what we have and what we want. I hope.
So you think you would be able to integrate additional levels of information
Yes,
into
we
the
have
same
right now
AI model?
Yes, we have multimodal AI these days, which can integrate different modalities.
That's what I was asking
Yeah,
you. Do you have a
exactly.
multimodal
Yes,
approach?
exactly. This is something that we are working on it right now on another project. Again, it's about HCC, and we are integrating different modalities. We have MRIs, and at the same time, histopathology, and we want to integrate different modalities. Yes, I can say it's possible, but of course, it has its own challenges. Yeah. Thank you.
Just for the sake of time, I think we'll end the questions there. So thank you very much again to our speakers and to everybody who asked a question. We'll now hand over to Tim Kendall and Kevin Gaston, who are going to lead the rapid fire science, right?
Thank you very much.